So I'm really pleased to introduce today's speaker, Emily Stevenson. Emily has recently submitted her PhD thesis from the University of Exeter and Plymouth Marine Laboratory, where she has spent the last 4 years exploring microbial communities of microplastics. In this talk titled Microplastics and Their Microbial Reefs, Emily will be discussing some of her findings so far. So without further delay, I'll hand over to today's speaker. Emily, over to you.
Easy. Thanks, Sophie. And it's really, really lovely to see you all. And I'm really looking forward to giving this talk. Comes at a really good time because I have literally just submitted my PhD thesis. So it should all be fresh in my brain. And my viva's in about 20 days. So it's all good practice. So try to be gentle with your questions, as I hope that I'll receive gentle questions in my viva. So, as you just heard, my name is Emily Stevenson, and I have just submitted my PhD at the University of Exeter with Plymouth Marine Labs. But before I dive into that, a little bit about me. I'm going to introduce myself. So, I'm really lucky. I get to wear lots and lots of different hats. I do get around quite a bit. As well as doing my PhD, I'm also the co-founder of a marine conservation organisation called Beach Guardian, and through that we empower communities to tackle plastic waste. So everything I do is about anthropogenic pollution. I'm really, really interested in human-derived waste in the marine environment and how all of the sort of sectors of animal health, human health, and environmental health are linked under this One Health concept. I've also done a bit of policy work. I worked as a senior policy intern at the House of Lords a couple of years ago. But as I said, today I'm really just going to focus on me as a researcher and what I've spent the last 4 years of my PhD doing, but also 2 years of my master's prior to that. So my PhD title was Microplastics as Vectors for Antimicrobial Resistance in Aquatic Systems. A bit of a mouthful and doesn't really mean a lot. So I'm going to go through some building blocks to help establish the foundation of background for my research. Not to insult any of your intelligence, I'm sure you're all aware of this, but just to make sure we're all on the same page before diving in to some more of the detailed stuff. So, bacteria. These are single-celled organisms, and everybody knows that bacteria are extremely important in the human body. We've got 10 times more bacterial cells than human cells in the average human body. But it's not just humans and animals where bacteria are important, but the environment too, especially the marine environment, with an estimated 1 billion bacterial cells per litre of seawater. And of course, a lot of these bacteria can be completely harmless, and this is what we call commensals. But we've also got pathogenic bacteria. These are the disease-causing bacteria, and they can be extremely damaging to biosecurity, human health, animal health, and so on. Now, one of the biggest challenges we have with bacteria and other microbial infections is drug resistance or antimicrobial resistance. When you hear me say AMR, which you'll probably hear me say a lot of, I'm talking about antimicrobial resistance. And this is the ability for microorganisms, so bacteria, viruses, fungi, and some parasites, to evolve the ability to multiply and persist in the presence of an antimicrobial, being an antibacterial, antibiotic, antiviral, antifungal, and so on. And just to sort of give you a visual demonstration of what that would look like, on the screen here, this is an agar plate, and you've probably all seen or heard of one of these before. You've got this nutrient-rich jelly, which allows bacteria to grow. That sort of slimy, snotty stuff you see in that plate, that's all bacteria. And those white discs, they're containing an antimicrobial. And you can see on the agar, it's completely clear around those discs, meaning that the bacteria have died. They haven't grown there, so they are not drug resistant. They are susceptible to those drugs. However, when you've got an antimicrobial resistant organism, it will grow in the presence of an antimicrobial. So you can see in the agar plate on the right here, we do have bacteria which are drug resistant, able to grow in the presence of those antimicrobials. And this is really, really damaging for some of the most basic infections not being able to be treated. And this is one of the greatest human health threats of our time. So now you're probably thinking, well, if it's a human health threat, where can microplastics come into this? And I'll get onto that. But just to sort of set the scene with microplastics, within this talk and within my research, I use the definition of microplastics being any plastics less than 5 millimeters in size. I know that this is up for debate, but just to set the scene, I use less than 5 millimeters. And microplastics are extremely prevalent. Whilst they may be small in size, they are certainly not small in quantity. With an estimated 125 trillion particles already accumulated in the aquatic environment. So vast, vast quantities. So where do these interactions take place between all of these building blocks that I've established? Well, when we look at the sources of microplastics, how do they get into the environment? It's incredibly varied. You've got land-based sources, you've got sewage-related sources, at-sea sources. And when we look at the sources of antimicrobials, they massively overlap with the sources of microplastics, meaning in many environments across the world, you've got microplastics, antibiotics, antimicrobial resistance genes, and human and animal pathogens existing in the same environment. They're all coexisting and interacting with one another. For example, take the sewage system. We know that this is a primary route that microplastics will enter the environment. We also know that this is a hotspot for antibiotics that haven't been degraded in the human body, and all of the microbes that are contained in our poo end up in this one system, so they're all interacting with one another. And that's where my research lies, is exploring those interactions between these co-contaminants. And the culmination of all of that leads us to the plastosphere. I love that name, the plastosphere. I think it sounds almost like something out of Star Wars, but that just, I'm just a nerd. The plastosphere is essentially these microbial communities that attach to plastic debris in the environment. So when any surface exists in the environment, biofilms will form. If you're a marine biologist, you know all about biofouling in the environment. Well, on a microscopic scale, these microbial biofilms will form on surfaces. But what's interesting about the plastosphere is that research has already shown that not only are these communities frequently distinct to the surrounding environment and other surfaces, but they could also be hotspots for pathogens and drug-resistant bacteria. Now, from this point, you might be thinking alarm bells are ringing. There could be, real cause for concern if that is the case. And that's what I've spent the last 4 years exploring. Now, here's some pictures to give you an idea of what I've been looking at. Obviously, this is not to scale. What you see here, these are microplastics. On the left, you've got a bio bead derived from the sewage environment. These are used as a filtration medium within sewage systems. And on the right there, you've got a nurdle. This is the pre-production pellet that's used in plastic production. So these are about 4 millimeters in size. And what you see here, these are not bacteria because you can't see that, but this is likely fungus growing on these microplastics. So just from these pictures, you can see how diverse these communities are, and how abundant these communities can be. Now, just in case I haven't explained that enough, I have a video here to show you, which sort of is a bit comical. I must sort of explain that this is copyrighted. This is not mine. This is straight from the BBC. This is actually from a Doctor Who episode I was watching one Saturday evening, and when you hear it, you can imagine my surprise when they started talking about this. So this is just a short video to sort of summarize some of my research.
The antibiotics aren't working. He's getting worse.
Doctor! Doc, Haramu said that the birds are getting really angry out there.
Not surprised if this is what their insides are like. Take a look. They're full of plastic.
Once plastic gets in the ocean, birds mistake it for food. I've seen hatchlings regurgitate 200 individual pieces.
It's feasting on the plastic in the birds and taking them over. The alien pathogens attacked the plastic. The plastic metastasized as if it was living. Autons? No, it can't be autons. They don't work like this. Not bacterial. Come on, brains, move it along.
She said brains, plural.
But how does that explain what happened to Jamila? Like, she just got taken over.
Yeah, how does it? It attacks the plastic in the birds, but there's no plastic in humans. Except... oh, microfiltration.
Hey, Doc, that's not a thought. That's just a random phrase.
No, that's a very big thought. The microfiltration system in the water. It filters out microplastics. Alien bacteria homing in on microplastics because humans are full of them.
Excuse me, I'm not full of plastic.
Full of something.
I'm afraid you are, Graham. It's in the air, it's in your food, it's in your water. Humans have flooded this planet with plastics that can't be fully broken down. So much so that you're ingesting microparticles whether you know it or not. You're poisoning yourselves as well as your planet. An alien bacteria has come to this planet and it found a feast.
Now, obviously, there is some very cringeworthy acting in that clip. And of course, I'm not looking at alien bacteria on plastic. I'm looking at, you know, human-associated bacteria on plastic. But I think this really exemplifies how all of these things can be very easily linked, how the threat of bacteria on plastics isn't just an environmental threat, but that plastic is getting into the body of animals. That plastic is getting into humans. So could plastics be a vector for these microbial communities that are growing on them in the environment? And that is the sort of thing that we're interested in researching. Let's not hear that again. So now into what I've actually been doing and some of the research we've been conducting, some of the findings we've been generating. When I started my PhD, I had three things I really wanted to look at. And that was, are plastics selectively colonized by bad bacteria? And by that I mean, do the drug-resistant pathogens choose to colonize plastics over other substrates that exist in the environment? Because we know that biofilms have a role in drug resistance. We know that biofilms have a role in supporting pathogens. So are plastics unique in their ability to support these communities? The second thing that I'm interested in is looking at whether there are unique properties of plastics which could influence the evolution of drug resistance. And then finally, could plastics in the ocean act as a vector for antimicrobial resistance, either between environments or within food chains? So let's look at this first one. And these were the experiments that I did in the first year of my PhD. Really simple experiment where we basically took 5 different particle types. So nurdles, these are the pre-production pellets. Biobeads, these are microplastics used in sewage. Polystyrene, we all know what that is, wood and glass. We sterilised these particles, we made sure they were all the same size, shape, and we put them into microcosms with a natural sewage community and allowed biofilms to grow on the particles. And then simply, once the biofilms had grown after a set time frame, we removed the biofilms and essentially looked at what grew where, so that we could see if certain particles would choose for the more harmful bacteria, in very simple terms. So this is just a summary of my results, and please, if you're interested, check out our paper in microbiology. This is looking at resistance gene prevalence. So we chose a resistance gene marker. It's widely used as a proxy for anthropogenic pollution and AMR in Taiwan. That's very sort of specific detail. Essentially what we're looking for is the proportion of these communities that are carrying this resistance gene. And on the plot here, you've got our different communities along the bottom. So the liquid, the planktonic community, glass, wood, biobeads, nurdle, and polystyrene. And what we see here is that wood and polystyrene communities harboured a greater prevalence of this resistance gene marker. So wood and polystyrene in this experiment were found to be important for selecting for antimicrobial resistance. So from this, we start to think, okay, there are particle-specific drivers which are involved in the communities that are attaching to these particles. And then we also wanted to look at pathogens, because antimicrobial resistance genes in commensals is a problem because they can spread to pathogens, but antimicrobial resistance genes in pathogens is even more of a problem because they're the ones that cause the disease. So this is looking at E. coli, which are more likely to cause infection. And what we found was that the biobead communities had a significantly greater proportion of E. coli that were pathogenic. This to me was a big concern because biobeads, as I've said, are used in sewage. They're used to clean sewage. They're used to encourage the formation of biofilms. And as we know, in sewage, it's full of E. coli because that's coming from our gut. So if the worse E. coli are attaching to biobeads and then they spill into the environment, that could have real consequences on surrounding waters, people using them for recreation, and also any organisms feeding in that area. So from that first experiment, we're like, okay, potentially there are substrate-specific drivers influencing colonization. And I'm sort of going to go not in chronological order here, because then we went to this question: are microplastics vectors for antimicrobial resistance? And as I said, there are two things that we're interested in here. The first is, we know microplastics are persistent. They do not degrade. We know that they are buoyant for at least some time in their oceanic life cycle, so they can be transported. So if these communities are colonizing microplastics, are they spreading into different environments, maybe pristine or novel environments? The second concern is that we know microplastics are ingested by a whole host of marine organisms. We also know that if a microplastic has a biofilm, it sort of masks the anthropogenic smell of the plastic, so it's more likely to be eaten. So could microplastics carry antimicrobial resistant pathogenic bacteria into the food chain, and then is there a consequence on us being at the top of the food chain? So let's look at that first one. So we wanted to go into the environment. Let's get out of the lab. Let's see if these findings are reproduced in the environment. So what we did is we took those 5 particle types from the first experiment, because it's really important to not just look at plastics, but those control particles as well, to really answer that are plastics unique question. And we made these structures that you can see on the screen here to allow our particles to be free floating, because we didn't want to put them in cages, because the cages would grow a biofilm and it would influence the biofilms growing on our particles. So we had these structures and we put them in 4 different locations in Truro in southwest England. The first location was a hospital sewage tank. This is untreated clinical waste at Treliske Hospital. It was foul, but so cool to go and see that and had put my particles in place, obviously with lots of PPE. We then sort of had a transect moving downstream from the hospital site. So then our next structures went in place upstream of the wastewater treatment plant that receives that waste, as well as domestic waste. And then a downstream site from that wastewater treatment plant. And then finally, an offshore sort of marine site. So sort of looking at how the communities change as a variation of anthropogenic pollution. And we had those biobeads. And what's important about this site is that in 2010, 5 billion biobeads spilt from that wastewater treatment plant. So these biobeads are of ecological relevance to this system because they're all still there 15 years later. And those nurdles again. And we left them there for 2 months to allow the biofilms to grow. And what we did after that 2 months is we took the particles out, we extracted the DNA, and then we sent the DNA for metagenomic sequencing and ran it through a pipeline which would allow us to investigate the antimicrobial resistance genes in these samples, as well as the community that is present within these samples. So similar to experiment 1, we're looking at what's growing where, but this time we're looking at the influence of environment as well as substrate. Now, this is currently under review, so I'd appreciate if you don't take any pictures of this just yet. Hopefully within the next month or so, this will be out and you can do whatever you want with this data and share it as widely as you possibly can. But what this heat map shows is you've got each sort of environment is separated. So you've got hospital going down upstream, downstream and marine. On at the bottom there, you've got our different communities. And then this is separated by resistance gene class. And this data is standardized. This is read counts of antimicrobial resistance gene standardized to total bacterial reads. So this is giving a proportion of the community that we had a positive read for these resistance genes. And what is interesting is we do seem to see an influence of environment, but some resistance genes decrease in prevalence moving downstream. As you would expect, the hospital is full of a whole load of contaminants. So you would expect to see that there's a lot of resistance genes in there. And if I just go back, you can see there's a lot of resistance genes present in the hospital, and that's shown by the lighter colours moving to red. That's a higher proportion of resistance genes present. So at the hospital, there's loads of stuff, and as you move downstream, it gets darker, so there's less. So looking at fluoroquinolones, for example, there's more in the hospital than there are in the marine environment across particle types. Expected. However, some resistance genes did increase in prevalence moving downstream. So looking at aminoglycosides, you can see that the colours actually get lighter moving downstream, which means there's a greater proportion of resistance genes in those samples downstream than upstream. And interestingly, we do start to see an influence of particle, and some particles were highlighted to pose a greater AMR risk. For example, looking at nurdles and polystyrene in both the downstream and marine environment, we have a really, really high relative abundance of oxazolidinone resistance genes, and we don't see that in our other particles. So similar to our first experiment, there could be substrate-specific drivers supporting antimicrobial resistance enrichment. So moving on from AMR, we also wanted to look at pathogens, as we did in the first experiment. And what we did is we matched our taxonomy data with a published list of bacterial pathogens known to infect humans. So we could see how many of our how many of our species that we found were known to be human pathogens. And out of 1,223 pathogens in this database, we found 641 across our samples. And I'll show you a very similar plot to before, but now we're looking at pathogen prevalence. This is pathogens standardized to the size of the community. And what you can see, again, we have this influence of environment, where in the hospital we have more pathogens, as you would expect. And like we saw with the AMR data, some pathogens decrease in prevalence moving downstream, but some pathogens increase in prevalence moving downstream. I understand that this is hard to see, so let's zoom in on one. This is flavobacteria, and this is all of the different species of flavobacteria. And what you can see very, very clearly when we look at a species level is that as you're moving downstream into the marine environment, you're getting way more colours, way more hits, and this is an increase in prevalence of these pathogens. And what I think is particularly stark in this figure is that if you look closely in upstream, downstream and marine, all of our particles have a much brighter colour than the free living. That's the seawater sample. So it looks like those pathogens are not really present free living. They're becoming enriched in our particles. And I found that was particularly interesting and important. So just to summarise that, because I know I showed you a lot there, what we found with this study was that environmental parameters are more selective than particle-specific characteristics. And we have done statistical testing on this using PCOA plots, which will be in the publication. So environmental parameters were significantly influencing both community, taxonomy, and antimicrobial resistance. But many resistance genes and pathogenic species of concern did increase in prevalence within the marine environment, which is really concerning because that goes against our traditional understanding of how surface waters are not as contaminated as super contaminated waters. We're not really super concerned about surface waters because we think that it is clean and safe, but actually what we're seeing here is actually there are some resistance genes that we really should be monitoring in surface waters. And what I was pleased to see going against reviewer 2 in my first experiment, this experiment did validate the lab-based findings that polystyrene does seem to be important when supporting antimicrobial resistant bacteria. And finally, the sort of take-home message, I suppose, from that study and the one prior is that these findings do support the concept that the plastisphere is a novel environment where resistance genes and pathogens can be enriched. And even if they're not enriched, they're present. They are definitely there in the environment, and we know that there is this threat posed by plastics that they don't degrade. So moving on to the next thing, and this was a really fun experiment, looking at how microplastics could be a vector for these communities in the food chain. So what we did was we wanted to sort of explore a relevant model system. We know that mussels used in aquaculture really good at filtering out pollutants, not just microplastics, but bacteria, antibiotics, and that kind of thing. So what we did is we took mussels from the environment, we gave them a period to acclimate to the lab system and depurate their environmental communities from their gut. And then we exposed them to various different treatments. A treatment of sterile microfibers, because that's relevant for the sewage environment. We exposed them to a sewage community. We exposed them to microplastics and sewage. And then a final treatment where we grew biofilms from the sewage on the microfibers and just exposed them to the colonized microfibers. And we exposed these mussels to these treatments for a period of 8 days. After the 8-day period of exposure, we extracted their gut, their poo and the water, extracted the DNA and ran qPCR on a set amount of gene targets so that we could see whether resistance genes were accumulating in the mussel gut as a result of ingesting microplastics that could carry, like a Trojan horse, these communities from the sewage into the mussel. And what we found was a surprise. So I'm just going to show you one plot from this in the interest of time, but also this is the most interesting thing that we found. So this is going back to the INTI1 gene marker that we used in the first experiment. This is a marker for a proxy for antimicrobial resistance. This box plot simply shows on the bottom there, you've got pre-acclimation mussels, post-acclimation, our control group that weren't exposed to anything, our group that was exposed only to microfibers, so there was nothing in there, just microfibers. Then the sewage alone community, inoculated microfibers, and sewage and microfibers. And what took me by surprise was that we saw a significantly greater prevalence of this resistance gene in the microplastic alone treatment compared to our 3 controls. So there was something about adding in microfibers that led to the retention of resistance genes in the mussel gut. And what we've sort of derived from this is that when there is also a bacterial community present, like this contamination from sewage, those bacteria trigger the immune response of the mussel, which sort of decimates the gut microbiome. However, these stealthy microfibers that are in there alone, that's not enough to trigger the immune response at the concentrations we used. And so they get retained, and then the bacteria that are already present in that gut community attach to the microfibers in vivo, form these biofilms, and start exchanging resistance genes in vivo, because there were already bacteria harboring INTI1 in the seawater. It's just absolutely everywhere at this point. So that's where it came from. And just by adding in microfibers, we think it's providing a surface for biofilm formation in vivo. We need to do a lot more testing on this, but I just wanted to show you this interesting result just to get us started. Okay, and then finally, don't worry, I haven't got any data to show you on this one because we're still looking at it. With all of this in mind, are there any unique properties of microplastics that could influence the evolution of antimicrobial resistance? And we wrote a review at the start of my PhD looking at all of the potential things that have already been discussed and summarizing it into 3 hypotheses. There are 3 ways that microplastics could be influencing the evolution of AMR. And that is, microplastics provide a surface for the exchange of genetic material, but this is seen in other biofilms. They also act like a magnet for other co-contaminants, so antibiotics and heavy metals adsorb to the surface of the microplastic, which could influence evolution of AMR. But it's this third one over here that I am super interested in. This is what makes plastics unique. When we make plastics, we put thousands and thousands of different types of chemicals into them. And these chemicals are UV stabilizers, plasticizers, antifoulants, all of these different chemicals that enhance the properties of plastics. And some of these are known antimicrobials. Simple concept of natural selection. If you've got a selection pressure present, it could lead to the evolution of a response. Like if you've got an antimicrobial present, it could lead to the evolution of antimicrobial resistance. So at the end of my PhD, I went to Australia and I did a placement with Jake O'Brien and Kevin Thomas to look at these chemicals, these plastic additives, and to see if they could influence the evolution of AMR. I'm going to leave that as a cliffhanger because I don't want to give any spoilers, but we've got some really interesting findings so far. But you know, it really is those chemicals in plastics that are super interesting when looking at these communities. Now finally, this was what I ended on with my PhD. I started thinking, well, bloody hell, I've gone super niche with this, as does any researcher. I'm looking at specific communities in a specific environment with these specific particles. Can we think bigger? We've already linked climate change and plastic pollution. On a sort of global scale, we know that climate change and plastic pollution are interlinked. We've also linked antimicrobial resistance and climate change. We know that they are two interlinked things. And by interlinked, I mean they're sort of synergies. They encourage one another. But looking at this, I'm like, oh my goodness, we're missing something here. We haven't done the link between plastics and antimicrobial resistance, other than the communities that are present on them. So we took a systematic approach and we looked at the whole cycle of plastic raw material extraction, production and manufacture, use, collection and disposal. And we thought, do any of these points influence antimicrobial resistance, the whole antimicrobial resistance crisis that we're facing? And what we found, and this is published, is that at every stage, there was something that would influence antimicrobial resistance, linking those two planetary threats of plastics and antimicrobial resistance. So I really wanted to zoom out and see what was going on from a systems approach, and I really enjoyed doing this paper and making this figure. So that's sort of everything that I've been getting up to for the last 4 years. Again, thank you very much for the opportunity to come and talk to you guys. And I'll very happily take any questions. And I'm looking for a postdoc, so hit me up if you know anything. Thank you.
Brilliant. Thank you so much, Emily, for a really interesting talk. We've had a few questions come in, so I'll just start running through those. So first off, someone has asked, did you take into consideration any temporal factors that may have influenced results, i.e. time of year, climate, water temperature, water level, etc.?
Yes, it absolutely would have influenced our results. So we did the environmental experiment from February to April. We wanted to try and get in before the bathing season to see what was going into our waterways. If we did the experiment over a longer time period or at different points of the year, we would see different results, I'm sure. Other studies have seen an influence of season, whether that be temperature or changes in salinity and that kind of thing. It's extremely complex. So absolutely, that would influence it. We did take into consideration temperature because each environment was a different temperature. The wastewater system in the hospital was very different to the temperature in our marine system, and even in that small scale we saw an influence. So temperature definitely has an impact on these communities.
Brilliant, thank you. Next question has asked, if you had unlimited time and resource, what else would you have explored as part of this PhD project?
I'm already tired enough from what I did. No, I'm joking. If I had unlimited time and resource, I would really love to do this in different parts of the world, because you can't derive huge sweeping statements that you can generalize across every environmental system from one environmental study. So if I had unlimited resources, I would do a similar study, but in every continent and in every season across 5 years, with lots and lots and lots of replicates and all the money in the world to do as much metagenomic sequencing and pay for someone smarter than me to do more elaborate analysis on the metagenomics. And then we can really start to pull out definites, or at least close to definites, instead of the maybe, possibly be.
Brilliant. You're right. That does sound exhausting. So someone else has asked, are there any management actions that can be taken beyond simply addressing plastic pollution at the source?
Yes. So this is what I want to move on to next. And that is, I believe, have a low-hanging fruit solution available to us. And that is looking at these wastewater systems where we know plastics are there. We also know that plastics are intentionally added to these systems as filtration devices. We know that antimicrobials are there. We know that human pathogens are there. And we know that that is all spilling into the environment. So what we need to do is look at these systems and not say, don't ever use plastic, it is the worst thing ever, but really investigate what is the least damaging plastic in terms of antimicrobial resistance and pathogen burden within those systems. And that's what I want to do. And that's sort of the direction I'm taking my research, is looking within the wastewater industry of how we can make the least changes to infrastructure, because, it's expensive, that have the biggest positive influence in reducing the amount of resistance genes, pathogens and microplastics going into the environment. So yeah, I think it's down to wastewater systems where we need to be making changes.
Brilliant, thank you. Next question asks, were there any beneficial organisms or physiochemical processes you found during your research that are worth exploring more?
Oh, absolutely. I love this question. So it's not my specialty. There are lots of other people that do this a lot better than I do. But we know that in these communities, plastic degraders are present. We know that there are bacteria living on microplastics that are able to use them as a carbon source, and that will actually break down the plastics completely. So I had a couple of master's students in my final year where we looked at plastics we'd collected on beach cleans to see if there was any plastic degraders there. And there is an abundance of plastic degraders in the environment. The problem is they're not that good at doing it. They take ages to actually sort of reduce the weight and size of plastic to a significant amount. So what we need to do is some evolution and manipulate those bacteria so that they can be super degraders and sort of start degrading plastic over a timeline of months instead of centuries.
Fantastic, thank you. That's really the end of our questions. One person has said if this presentation could be shared, that would be amazing. If whoever left that is still on the call, I would say please email the Science Talks inbox. We will be sharing this internally, but I think given there was some unpublished material on some of the slides, it might take a while for it to be shared more publicly. But yeah, thank you again, Emily. That was fantastic. So I think we'll leave it there.
Thanks. Well, thanks everybody.